Okay, thanks. So I'd like to first thank the organizers for the invitation here. I'm going to talk about something maybe a little bit different from that title, but definitely thinking along the lines about how the nervous system evolves. And I'm going to focus on, again, thinking about this in an evolutionary context. And like many evolutionary biologists, of course, what we're fascinated by is how evolution has generated an incredible diversity of organisms. And my lab's been very interested in understanding how evolution's generated this sort of morphological diversity, and this obviously just gives you a snapshot and just within some groups of animals about that kind of diversity. And we've focused now for quite a while on trying to understand this evolution within arthropods. And there are a couple of reasons that arthropods have proved really useful for this. One is that within arthropods is Drosophila melanogaster, which is one of the key sort of genetic model systems for understanding development in great detail. So many of the genes that we've identified that play important roles in development have been identified in that organism. So we can use that sort of as a launching pad sometimes for a lot of our studies. But the other reason, of course, is that arthropods are a remarkably successful group of organisms. I mean, one could argue in terms of species diversity and in some sense in terms of morphological diversity, they really sort of represent the sort of one of the great extents of evolution of being able to generate that kind of diversity. And I'm going to focus on a particular sort of, I think, important aspect, morphological aspect of arthropods. And in fact, it's where the name arthropod comes from. So they're jointed limbs, right? And that's one of the hallmarks of these animals, is that they have these appendages on virtually all of their segments, okay? And the beauty of these appendages is that they can be specialized to do many different things. So in some arthropods, the appendages can look virtually the same throughout the trunk. But in other animals, such as this mantis shrimp, you can have specialized appendages. And so if you've ever tried to catch a lobster without rubber bands on it, you kind of have an idea about the power of this, right? So the animal can actually do multiple things simultaneously. So it can attack you, it can try to run away, it can still be moving its antenna, it can do all these things at the same time. And that's because it has this incredible diversity of appendages, and then it has control of those appendages to do different things. So, for example, just in crustaceans, you have this incredible diversity of appendages. So that's not only diversity between species, but actually along the body plan of an individual species, you can have this great diversity. So one of the things that we're interested in, of course, is how you generate that kind of diversity and what the genetic molecular underpinnings are for that. But what I want to sort of emphasize here today a little bit, because this is a meeting about the evolution of the brain and behavior, is, of course, just changing the morphology of the appendage is really not enough. To make a functional segment, a functional appendage, there also has to be changes in the nervous system and changes in the musculature and things like that. And that's eventually what we'd like to explore. But I'm going to start by telling you sort of where we are with sort of thinking about this. So let me go back a little bit to neurogenesis. And there's an aspect, I think, of the way that the nervous system is built in arthropods that lends itself to being particularly easy to evolve these kinds of specializations of appendages as well. Okay, and this just illustrates two examples of insect nervous systems. So this is a grasshopper here, and this thing looks like a scale bar is Drosophila instead. And so for all the tiny size of flies, they're still really good for genetics, but obviously this makes it a little easier for me to illustrate what's going on. And one of the hallmarks of arthropod nervous systems is actually, we say they have a central nervous system, obviously. You can see the brain up here. But in fact, a lot of the nervous system is decentralized. There's a lot of function that's actually associated with nervous system that's within the segments, right? These will eventually be ganglia. Again, here we can go back to flies in a side view, and this is just a marker of neurons that you're seeing here. So the brain is over here, but you can see then the nerve cord going down the length of the animal here, the head's here, posterior, and then peripheral nervous system here. And let me, again, illustrate here with a little bit of a movie. So in this movie, the red are the neurons, and the blue is the axons. And again, this is actually then just looking at this CNS and not the brain. And so again, you've got actually this large number of neurons in every segment, so several hundred And these are actually where the motor neurons are, the controlling sort of local things within that segment. And so just as with in the CNS, then the peripheral nervous system, and then the muscle. So all of these things are repeated in these body segments. Okay? So you have this repetition of this entire pattern. And finally, just the motor neurons actually then going out to these muscles. In the case of flies, the nervous system sort of does get compressed up towards the front. But in most arthropods, then you have these distinct ganglia, even in the adults, that go along the length of the body plan. And again, if you look in any one of those neuromirrors, right, you actually have a very stereotyped development of cells within that. So you'll have very particular motor neurons born, particular interneurons. And this is just sort of a wiring diagram that applies both to grasshoppers and flies, and probably in general to probably both insects and crustaceans. And so you have this pattern, and it's repeated there over and over again. Okay? But the pattern is not identical in every segment. Okay? So there are specializations. Just as the limbs have differences between different segments, the nervous system also has that. And I just illustrate with a couple examples here. So for example, here's a particular type of cell type, and it's not important which ones right now. But you can see that this is the nerve core, the anterior here, and you can see that in abdominal segments A3 through A7, there's far fewer of this particular cell type than there are in other segments. It's not just that there are fewer in the abdomen or something like that. Here's another example. If you look at these more lateral cells here, you can see that there's more of them in abdominal segments and slightly few actually in thoracic segments. So there are all these segment-specific differences, and there are lots and lots of markers that illustrate this. Okay, so how do we think about these segment-specific differences in arthropods? What generates that? And of course, we understand really quite a lot about the genetics of that. And this just illustrates one of the famous examples of the kinds of transformations that you can make in Drosophila that change the identity of one segment into another, or at least in this case, the identity of one appendage into another. So in a normal fly, on the second thoracic segment, there are these wings. So you have a single pair of wings in this animal. And on the third thoracic segment, instead of a wing, there's this structure called a haltere, which is actually sort of more like a gyroscope that provides information to the fly as it's flying. And in a mutant in the gene called ultrabithorax, and this is not a null mutation, this is a mutation that just changes its regulation a little bit. If we eliminate that function in the developing structure that normally makes the haltere, that haltere then instead develops as a wing, and you get this remarkable transformation in the body plan where you can now change one appendage identity into another. And so it turns out, of course, that there's an entire family of these Hox genes, and this is probably one of the sort of iconic images in developmental biology. And they're in Drosophila, there are 8 of these, and they're organized along the chromosome in the same order that they're deployed along the axis of the animal. And their function is to regionalize the body plan, to define differences that go along that AP axis. And it turns out, of course, that we've learned about 20 years ago now that this family of genes is incredibly well conserved. And so, for example, you find that same family in mice and humans. There's some expansion. So, for example, in mice and humans, there's 4 copies of the complex instead of just 1. But again, within a copy of the complex, there again, the Hox genes are organized in this linear array that's also reflected in their expression down the axis of the animal. And various genetic studies now in many different animals indicate that they have a common function in regionalizing the body plan. Here's just a picture in Drosophila again, so anterior is here. And you're looking at 4 of the homeotic genes, the protein of them. And this thickened area here is the nervous system. And so you can see that not only are these Hox genes expressed in the ectoderm, where they're going to control the eventual formation of the ectodermally derived appendages, but they're also expressed through the nervous system. In fact, at later stages, they're more highly expressed in the nervous system than in other tissues. And in fact, they control that regionalization, those regional differences in the nervous system as well. Okay? So now what I want to do, though, is ask, well, do you actually use this system in evolution to change body plans of arthropods? Okay? So you're capable of doing that as a mutant in Drosophila. Okay? But is there any evidence that you actually can shift those boundaries in a meaningful way in organisms to actually generate this change? And in fact, the answer in insects, by and large, is that you don't. Right? So, for example, we can take a two-winged fly and turn it into a 4-winged insect that looks something like, say, a dragonfly. But in the end, people have shown that, in fact, that's not what goes on in insect evolution, that, in fact, the Hox gene boundaries are relatively static and non-moving in insects. And instead, what's happening is you're changing the way that downstream targets respond. But a number of years ago, we started exploring this issue in crustaceans. So they used to be called, or until the phylogenies actually for arthropods has undergone quite a number of revisions. This is one that I think that's reasonably accepted now. And in fact, insects and crustaceans now sort of form a single clade, with crustaceans really being paraphyletic. But anyway, within the crustacea, though, you find much more variation in body plans than you do in insects. And so we wanted to know, well, do Hox genes actually tell us something about how these body plans are evolving? And to do that, we also needed to develop a model species within the crustaceans, particularly so that we could do experimental manipulation. So I'm going to tell you a little bit about that today. So the animal that we've chosen to do a lot of detailed studies is this one, called Parhyala hawaiiensis. And so, as its name might imply, the type specimen was collected in Hawaii many, many years ago. But in fact, this animal has distribution all over the world in tropical water. So anytime you're on a beach and you see gray things hopping in the sand, it's this group of organisms, at least, these amphipod crustaceans. So they're reasonable size. They're probably a little bit, they're about 2 or 3 times the size of an adult Drosophila melanogaster. They're very, very easy to keep. In fact, there's an interesting story about how we isolated these animals. So at the time, Bill Brown was a graduate student in the lab, and he went to the Shedd Aquarium, and he actually didn't go to any tanks where they keep anything. He actually went to the filtration system of the tank, and these are the animals that live in the filters, and they just eat garbage, and nobody takes care of them, and they do very well. So they make for a very good lab rat sort of animal in that case. And so I'm going to come back to these in a minute and tell you about our ability now to actually use these as an experimental system. But let me tell you about the particular type of morphology then that we're interested in. So we started off with a particularly striking example of a segmental specialization that actually changed with evolution within the crustacea. And that had to do with appendages at the anterior part of the thorax. So if you look at something like this is a brine shrimp, Artemia. This is typical of a crustacean that has actually relatively little differentiation of appendages along the trunk of the animal. So these 11 pairs of appendages here in the trunk are all used for swimming, and they're kind of like oars, and the animal moves them sort of, all in synchrony together as a wave along its body. And if you were to pluck one of these off and not remember which segment it came from, just from its morphology, it would not be that easy to tell where you'd taken it from. So they're fairly homonymous looking segments. But again, you can contrast that to something like a lobster, where you have lots of these specialized appendages. But the ones we're going to pay attention to are actually the ones in the front of the thorax. And so here's an example of another crustacean called a mysid, and it helps to illustrate this particular specialization. So a scanning EM of the underside here. So the thoracic appendages here in T3 on back, they're typical kind of swimming legs that this animal has, okay? And they're used for locomotion through the water. This maxilla is one of the jaw apparatus segments, and you can see that this is actually a feeding appendage, so it's much smaller. It has crushing parts on the front of it. It's positioned in front of the mouth, and the animal uses that to crush food and move it into its mouth. But if you look at the first thoracic segment, instead of a typical kind of swimming leg on it, it actually has an appendage which has been called a maxilliped, or literally a jaw foot. So even though it's on the thorax, its morphology is much more like a jaw appendage. And functionally, that's what it's used for. It's not held out. It's not used to swim. It's actually up over the mouth, and it's another auxiliary appendage that's actually sort of a massive jaw part for crushing food and moving it in. So it functions much more like a jaw appendage than it does a traditional head jaw appendage than it would a locomotory thoracic appendage. And so it turns out if you look in crustacean evolution, and the red indicates here the position of maxillipeds in different crustacean groups, the thought is that the ancestral crustacean had a relatively homonymous body plan. All the segments were identical. But during the course of crustacean evolution, and in fact possibly independently in different lineages, then there was a specialization of anterior parts of the thorax to be actually used for feeding instead of locomotion, and they evolved into these maxilliped structures instead that you see. So you can have, depending on the species you look at, anywhere from 0 to 3 pairs of these maxillipeds. And a number of years ago, Michaela Savaroff and I showed that in fact there was a perfect correlation between the expression of a particular Hox gene, in this case the crustacean version of UBX, ultrabithorax, and the transition in the body plan where you go from the feeding type appendages to the locomotory type appendages. So that's the expression is shown in blue, and you can see that those appendages not expressing UBX then are this maxilliped type, and then that UBX marks that transition to the types that are used for locomotion usually. And that, of course, the expression of this Hox gene then precedes, though, the development of the appendage. That's very early, and then the appendage is developed a little bit later. And certainly, given what we know about the genetics from things like Drosophila in mice, it's totally reasonable to postulate that, in fact, that this gene is actually responsible for that kind of transition. But of course, the problem and the difficulty often run into with the sort of evolutionary developmental biology studies is that it's very nice to get these kinds of correlations, and obviously our techniques for looking at gene expression make that quite easy to do now. But it's difficult to move on from that and actually do the kind of manipulative experiments that you'd really like to do in these organisms to show that that's the case. But what I'm going to show you today is that, in fact, we can do that now after many years of effort. This is the point of having developed Parhyella as a system that we use in the lab. So just to remind you again, in the Parhyella embryo, UBX is expressed actually from T2 on back, and those are the locomotory legs, or actually they're clearly not the feeding appendages. And in this case, in fact, T1 is a maxilliped. Okay? So if our hypothesis is true, right, that UBX actually controls this transition in the body plan of these different types of appendages, then we'd expect that if we move UBX more anteriorly, we should be able to make an animal with more walking legs at the expense of feeding appendages. And if we can move it more posteriorly, we should be able to make an animal with more feeding appendages and fewer walking legs. Okay? And so we can do those kinds of experiments now. So one way that we can overexpress genes is actually to make animals transgenic. And so we use a Minos transposable element, which was first developed in Michaelis Averov's lab. And in this case, we use a heat shock promoter, so an inducible promoter that we can turn on just by gently warming up the animals. And we clone our gene of interest behind that, and then we can inject this in, and we can make transgenic animals. One of the neat things in Parhyella, and I won't try to explain the detail of how we do it, is we can actually also make half transgenic animals. So we can transform one half of the animal and not the other half of the animal. That gives us a really nice internal control. And here's an example then if we do that misexpression to UBX. So in a wild-type animal, again, T1 is the maxilliped, and the rest of the appendages are very different looking from that. So here in this animal now, this side is normal. It's a normal T1 appendage. But now you can see we've transformed the other side, and that's now a full-blown walking leg, actually typical of a T4 type of appendage where there's very high levels of UBX. And in fact, all of the head appendages now are transformed towards walking legs, including the antennae. So it fits with our hypothesis about what this gene does. It is, in fact, capable of making that transition. But probably in some ways more interesting to us would be the reverse, the loss of function experiment, to actually move UBX further back and see if we can make more of these feeding type appendages. Okay? And again, we can do this in Parhyella. In this case, we do it by siRNA injections. So here's wild-type UBX expression. If we inject an siRNA knockdown expression, you can see that here. It's typical of this RNA-type approach that we don't necessarily get complete knockdown in every animal, but actually that's quite useful. We can get a phenotypic series from doing that. So what happens to these embryos as they develop? So again, here's the wild-type appendages. Here's T1 and T2. So again, T1 is the maxilliped. It's a short appendage with these crushing parts towards the base of it. And T2, again, is this totally different type of appendage. In the case of Parhyella, it actually has small claws at the end, which is used to pick up food. And so if we knock down UBX and we look at this segment, what we can see is a series of transformations. This is probably one of the more severe ones. And you can see now that it's a shortened appendage, and now it has these structures down at the base of it that are used for crushing. Okay? We'll come back to a minute and ask, well, but that's a good morphological transition. What really happens to other aspects of this appendage? But anyway, so at that level, though, we can certainly transform these appendages, and we can do something similar to what evolution has done then. We can actually generate different numbers of maxillipeds by doing this kind of manipulation, suggesting that maybe evolution is actually able to use this as a mechanism. We certainly see that correlation between what we see between species, and now we can go into one of those species and actually make that transition. So this is quite different from the case in insects, where insects don't seem to be making these changes by moving around Hox genes, but it does seem that crustaceans actually do it that way. Okay, and maybe that's also why crustaceans actually have a greater diversity of segment specialization, because they're doing it very high in the hierarchy of genes controlling it, and they can make these major transitions. But of course, that distinction of feeding and locomotory appendages is not the only specialization that you see in a crustacean body plan. And what I want to quickly do is take you through some of the other specializations and tell you that, in fact, we probably can explain most all of them now. So this, again, is the Parhyala body plan. And in fact, even though it's a small crustacean, it actually has a really rich diversity of appendage types in it. And so here's just a little bit of a summary about the different ones. So I showed you T1 is the maxilliped. T2 and T3 are actually gnathopods, which are used for picking up food. And T4 and 5 are walking legs that face forward for when the animal wants to move forward. 6, 7, and 8 are walking legs as well, but they actually face the other direction, and they allow the animal to back up. A1, 2, and 3 are pleopods, and they're used when the animal wants to swim. And 4, 5, and 6 are uropods, and they're like kind of anchors that hold it down in the substrate. So it has these wonderful specializations, and of course then it uses them functionally for different things and moves these appendages in different ways. So we've actually gone through now and looked at all of the Hox genes in Parhyala. And so this is just sort of a summary of in situs. And you can see that there isn't anything particularly striking, I mean, surprising here. It's a strikingly beautiful pattern, but it's not surprising that you have this sort of nested pattern going down the anterior posterior axis of the animal. And then we can, look at that, put it on a diagram, and see how it corresponds to some of the morphologies. And again, compare it to this idea that maybe it explains many of these transitions. And it does, right? There's a really good correlation with certain of the specialized segments and the transition points. But do they really define that, okay? And do they do that? And we can now actually go and experimentally address that. So let me just go through a couple of the other Hox genes. So for example, SCR, which is here expressed in the posterior part of the head, when we knock down its expression, we can actually now transform the last jaw appendage, maxilla 2, into T1. So now we can make another maxilliped, but we can do it now at the expense of the normal jaw appendage. So we can get the transformation to go in the other way by knocking that down. The Hox gene abdominal A is expressed in the more posterior part of the thorax and the more anterior part of the abdomen. So again, the distinction here is that in A1 and on through A3, you have these pleopods, these very fan-like structures that the animal uses when it wants to swim, versus A4 on back, which are these uropods which anchor down into the substrate. And so if we knock down abdominal A, we can actually transform these pleopods into uropods. So we can make an animal that has fewer pleopods now, but more uropods. And then abdominal B is expressed here through the whole posterior abdomen region in this area here. And we knock this down, we get a particularly striking transformation. So again, the thorax has these big, long walking legs, as opposed to the abdominal pleopods, the more anterior segments of the abdomen. If we knock down abdominal A, then in fact we can make an animal that has now actually more walking legs on it instead of swimming legs. So we can make all of these different types of transformations. And in fact, if we summarize all our data, I've gone through most of it except probably Antennapedia, we can now actually transform any one adjacent segment into another adjacent segment. And so what that means, if we think about it, is that if you look at the variation that you can get in crustaceans in terms of segment specialization, we now actually can give you a hypothesis as to how you would transform any one crustacean body plan into another crustacean body plan. So during evolution, we could probably define for you what the pattern of shifts of Hox genes would have to be. Okay? I showed you that our prediction, in fact, we did it the other way around for UBX. We started with the correlation and moved to function. Here we're actually defining their phenotypes, and now actually we can go the other direction and say, well, do the Hox gene patterns really look like that in other crustaceans? And that's something that we're starting to do, but I'll illustrate with one example that we can already say it's true. So again, in the abdomen of Parhyala, you have the 3 pleopods and the 3 uropods, and this is the pattern of abdominal A and abdominal B, and we can shift the relative number of each of these types of appendages by moving abdominal A and abdominal B. Okay? But there is actually a natural variation in this. So if you look at something like a crayfish instead of an amphipod crustacean, so a crayfish also has 6 abdominal segments, but it has 5 pairs of pleopods and only 1 pair of uropods. Okay? So that would lead to an obvious prediction for us, that if we move abdominal A back, that's exactly what we should do. We should change that. Okay? In this case, we don't even have to do the experiment because it's already published. So Tom Kaufman's lab many years ago published that, in fact, that's the expression pattern for abdominal A in crayfish. So it makes perfect sense that, in fact, these animals do actually seem to be doing this at the level of Hox genes. We don't yet know if they're doing it at the level of the regulatory elements of the Hox genes, or if they're doing this actually downstream, upstream of those genes right now, okay, where the actual genetic changes are. For various reasons, we think it actually is at the level of Hox genes, and we're actually developing techniques to be able to test these entire regulatory elements and see if that's actually how they're doing them how they're doing that in crustaceans. Okay, and so, but that only changes, and this is sort of getting now to other aspects of what would be going on, because obviously we've been very interested in just switching this morphology of the appendage. But if evolution is actually going to use this as a useful way to make variation, these appendages actually have to be functional all the time, right? And the end result has to be functional. So I showed you, for example, Ed Lewis's four-winged flies, right? And students, whenever you show that in a class, for example, in an introductory developmental biology class, students always want to know, well, does that fly fly? In fact, it doesn't fly at all. And that's because you've only transformed the ectodermal component of that wing. So it's just the halter that you've made the transformation, and you get a structural wing. But in fact, the nervous system that's underlying that and the musculature underlying that are not transformed. So it's actually not a functional transformation. So the question is, can in fact crustaceans actually make these functional transformations? Can we look at the way we regulate these Hox genes and understand how it is that you might actually be able to make this smoothly and actually make functional appendages the whole time that you're moving around Hox gene boundaries? And I should also state that you don't have to move the boundary abruptly. There's plenty of examples of crustaceans that have appendages of kind of an intermediate morphology. And we've actually seen in those animals you have modulated levels of expression and sometimes mosaic patterns of expression of Hox genes. So you can imagine making these transitions slowly. But however you do that, one of the things that you have to pay attention to is that the appendage is more than just that ectodermal structure. To be functional, it needs a proper musculature inside of it, and it also needs to be properly wired up to the nervous system. So is there a way, does this mechanism of using Hox gene regulation in some way lend itself easily to making all of those structures transform simultaneously? And this is really something now that we'd like to address. And so here, for example, I'm just showing you the musculature inside these appendages. We can make this, in this case, actually, it's a transgenic animal that expresses DS-RED in all of its muscle, and it gives us a very nice way of looking at live animals even and watching the muscle develop. And so you can see that there's distinctly different muscle patterns here in these appendages, and we've mapped we like to map these out and understand functionally, biomechanically, how they actually work these appendages. So the neat thing, and we'll get to, and we're starting to do these experiments, one of the nice things in Parhyala is that it also has a very unique, distinct early lineage pattern. And we've done a lot of work on this, which I'm not going to talk about, but just sort of summarize here. So it actually has this 8-cell stage embryo where 3 of these cells, these 3 macromeres here, lead to all of the ectoderm, sort of anterior, left, right, and posterior. This cell makes visceral mesoderm. And then these micromeres make germline, endoderm, and left and right somatic mesoderm. But what that means is that we can independently manipulate each of these germ layers during development. And so we've shown in other control experiments for other things that we're doing that we can actually restrict our siRNA knockdowns or our transgenic injections to individual blastomeres and then restrict what tissues are transformed. So one of the things we like to do, for example, now is knock down UBX just in the mesoderm and not in the ectoderm, and vice versa, and ask what happens. We haven't done the experiments yet, and we're gearing up to do those kinds of things. But I'll actually tell you that our bias, at least my bias, is right now is that the mesoderm actually is very naive, and it just pays attention to the morphology of the ectodermal segment that it runs into. Part of the reason that we say that is that, in fact, we've already done experiments where we've made mesoderm-free animals. So we've killed off all the mesoderm precursors in an animal. And the animal will get to hatching and will actually make a very good-looking Parhyala. It just has no mesoderm, no muscle inside of it, but the appendages look totally normal. So the appendages certainly don't need the mesoderm to develop. And we actually think that the mesoderm moves in and it actually gets its information. In fact, we've done other experiments showing that the segmentation of the mesoderm is very much dependent on the segmentation of the ectoderm. So that might solve one part of the problem very easily, that mesoderm actually just pays attention to the morphology of the appendage that it runs into. But the other thing, of course, is the nervous system and how that's wired, and gets back to the issues about evolving the nervous system. So if you're going to make these segment specializations, they're only useful if, in fact, there's an underlying wiring that is useful to that appendage, right? It makes the proper motions, holds the appendage in the right way. So I should say that all of these transformations that we've been doing, because we've been using an RNAi type approach, we're probably not knocking down these genes completely. But that's actually really good, because if we did, they probably would die as embryos, and we wouldn't actually get anything out that we could assay. But in fact, we get plenty of animals out that actually survive that have these modified appendages, and we can look at how they behave. And we're getting really interested in that. We know that, like in the case of UBX, where we transformed them to maxillipeds, that they actually do hold those appendages up over their mouth. Whether they move them in any useful way is something now that we'd really like to do. Our background is, or my background is as a developmental biologist, so trying to think about adult animals and how they move things is new. But of course, it's very fascinating, especially, again, from an evolutionary viewpoint. But what about the underlying nervous system? So one of the things that you can see in all of these is that, in fact, these Hox genes are not only expressed in the limbs, for example, but all through, in fact, those corresponding neuromeres. And again, we know in Drosophila that when you move Hox gene boundaries, it's not only ectodermal components that gets transformed, but it's the nervous system that gets transformed. So you change those segment specializations. And so we've been looking at this in Parhyala, and I'll just show you a couple of examples. So for example, antennapedia. And you can see at late stages, again, just like in Drosophila, very strong regionalized expression in the nervous system. And here for UBX, again, not only in the limbs, but through the neuromeres. And because we use this siRNA approach, we're not just knocking down in the appendages, we're actually knocking down throughout the animal. And so we think that what we would get is sort of a global reorganization of the segment, not just of the ectodermal components, but all of that. And so, but I'm going to leave you really essentially with that, because that's sort of the direction that we want to go now. So what we want to do now is define, well, what are the segment specializations within any of these appendages that allow for the proper use of an appendage. And of course, there's a great literature from crustacean neurobiology about those kind of control circuits and things like that. And we need to adapt that to Parhyala and then ask, in fact, if we get these kind of functional transformations in these animals. And that's really sort of the future of where we're going. So I haven't told you so much necessarily about the results so far, but I really wanted to leave you with a sense that here we have a system where we can really now do these experiments in a meaningful way. And so we'd really like to approach this now. And that's where I'll leave you. So I just want to acknowledge the people that did this work. So the initial work with spaces between names have disappeared. Anyway, the initial work on UBX was done by Danielle Lubicic, and then much of the rest of the Hox gene work that I told you about was done by Julia Serrano in the lab. And it's being continued by Heather Bruce and Erin Jarvis. And Erin is here at the meeting, and in fact, she's the one most interested in really looking at the behavioral aspects of these transformed appendages and then what's going on in the nervous system. And I'm going to go ahead and end there and take any questions. Thank you.